Welcome to Mythos AI, your source for Cliff's Notes on complex subjects. Join Anna and Phil for yet another engaging conversation.
Today we're diving deep into the world of AlphaFold 3, AF3 for short, a quite a significant advancement in biomolecular interaction prediction.
It builds on the successes of its predecessors, AlphaFold 1 and 2, which really revolutionized protein structure prediction. But, there were some limitations.
They had their limitations.
Yeah,
And those limitations are important to acknowledge as we delve into what makes AF3 so groundbreaking.
absolutely. The main weakness was in accurately predicting the interactions between diverse biomolecules, think proteins, nucleic acids like DNA and RNA, ligands, ions, modified residues, all these different players in the complex world of cells.
And AF3 steps in to address these limitations, offering a significant leap forward.
That's right. It can predict structures for complexes encompassing nearly all those molecule types.
It's like it's able to see the bigger picture, see the intricate dance of these molecules in a way that previous models couldn't quite grasp.
Exactly, and that's largely due to the innovative architecture and training methodologies employed in AF3. It's not just an incremental improvement, it's a fundamental shift in how we approach this problem.
Let's break down that architecture then, because I'm really curious about what's happening under the hood.
Sure, at first glance, the overall architecture might seem familiar, especially if you're acquainted with AlphaFold 2, but there are some key distinctions.
One of the key changes I understand is in how AF3 processes multiple sequence alignments, or MSAs, which are crucial for understanding evolutionary relationships between proteins.
Absolutely. AF3 replaces that computationally intensive evoformer module used in AlphaFold 2 with a more streamlined pairformer module.
Okay, so it's like they've swapped out a gas-guzzling engine for a more fuel-efficient model.
That's a great way to put it. This simplification reduces the computational burden without sacrificing accuracy. It makes AF3 much more efficient, especially when dealing with large, complex structures.
And then there's this novel diffusion module, which is completely new to AF3, right?
Right. This is where things get really interesting. Unlike previous models that predicted structures at the amino acid level, AF3's diffusion module directly predicts atom coordinates.
So it's a more granular approach, zooming in on the individual atoms that make up these complex molecules.
Precisely. And this has several advantages. First, it eliminates the need for amino acid-specific frameworks, which can be quite complex. And second, it avoids the need for what we call stereochemical violation penalties.
So it's a more elegant and direct solution.
In a way, yes. But of course, as with any generative model, and AF3 is no exception, there's always the risk of what we call hallucination.
Hallucination, in the sense that the model might generate plausible-looking structures, but structures that don't actually exist in reality.
Exactly. And this tends to occur more frequently in regions of a protein that are inherently unstructured or highly flexible. The researchers tackled this challenge using a clever technique called cross-distillation from AlphaFold multimer.
So they essentially had AF3 learn from a model known to be more reliable in those tricky, unstructured regions.
That's the gist of it. By training AF3 on structures predicted by AlphaFold multimer, which generally represents unstructured regions as extended loops, they were able to guide AF3 away from creating overly compact or improbable structures in those regions.
Reducing those hallucinations, so to speak.
Precisely. Though it's important to note that this doesn't eliminate them entirely. No model is perfect, and there will always be edge cases where the model might generate something unexpected.
Right, but it sounds like they've made significant strides in mitigating one of the main challenges of working with these generative models.
They have, and they've also gone to great lengths to ensure users have a sense of how much they can trust a given prediction.
You're talking about the confidence measures incorporated into AF3.
Exactly. AF3 provides users with valuable insights into the reliability of its predictions. These measures, such as PLDDT and PAE, are designed to give users an idea of how confident the model is in its output.
It's like having a built-in bullshit detector.
Kind of. It helps distinguish the more reliable predictions from those that might require a closer look or further validation. And the research shows that these measures are pretty darn good.
You're saying there's a strong correlation between these confidence scores and the actual accuracy of AF3's predictions.
Yes, and that's crucial, especially when we're talking about applying AF3 to real-world problems in fields like drug discovery, where accuracy is paramount.
Right, because what good is a prediction if you don't have a sense of how much you can rely on it?
Exactly. These confidence measures add a crucial layer of trustworthiness to AF3's predictions.
So we've got this robust architecture, a clever training process, these built-in confidence measures, but how does it actually perform? How do those predictions stack up against the real world?
That's the million-dollar question, and the answer is, well, quite impressively. AF3's performance has been rigorously evaluated, and the results are, to put it mildly, impressive.
The research supporting AF3's claims is quite expensive. Can you elaborate on how they went about evaluating its performance?
They used a variety of benchmarks comparing AF3's predictions against experimental data, the gold standard, and also against the performance of other leading tools in the field.
So it's not just about showing that AF3 can generate plausible structures, but that it can do so with an accuracy that rivals or even surpasses what we can achieve using other methods.
Exactly, and they didn't go easy on AF3 either. One benchmark in particular stands out, what they call the Posebusters dataset.
Posebusters, why is this dataset significant?
Well, it's a particularly challenging dataset because it's comprised of protein ligand structures that were released relatively recently, meaning AF3 wouldn't have encountered them during its training.
So it's like a real-world test. You're throwing new data at AF3, data it's never seen before, and seeing how well it generalizes.
That's the idea, and the results are, well, they're quite impressive.
So how'd it do? Did it pass the test?
It exceeded expectations, actually. AF3 significantly outperformed even the most advanced docking tools on this Posebusters dataset.
That's quite a statement, given that these other tools are specifically designed for predicting protein-ligand interactions.
And here's the kicker. It achieved this level of accuracy without any prior knowledge of the protein's binding site.
So it's doing true blind docking here, which means it has to figure out where that ligand binds based solely on the structures of the protein and the ligand themselves.
That's right. No hand-holding, no hints. It's like trying to find a specific grain of sand on a beach, but instead of visual cues, you only have, say, the feel of the sand beneath your fingers.
And AF3 manages to find that grain of sand, so to speak, with remarkable accuracy.
It does, which suggests that this more generalized approach that AF3 takes, this ability to learn from a wider range of data, might actually be a game changer for predicting protein-ligand interactions.
So it's not just about this one data set, right? They tested AF3 on other types of biomolecular complexes as well.
Absolutely. They wanted to see how well it handled different types of interactions. For example, they tested it on protein nucleic acid interactions, which are, of course, fundamental to life.
DNA replication, gene expression, these are all processes that rely on these intricate interactions between proteins and nucleic acids.
And again, AF3 demonstrated superior accuracy compared to more specialized tools designed specifically for these types of interactions, especially when you look at the larger, more complex structures. It even performed well on COSP15 RNA targets, which are notoriously difficult to predict.
So even when you throw those highly structured RNA molecules with all their loops and folds at AF3, it still holds its own.
It does, though it's worth noting that, at least for now, there's still some room for improvement in this area. For the really complex RNA structures, the research suggests that AF3, while good, doesn't yet surpass the accuracy achieved by human experts.
Which, to be fair, is understandable. Those RNA structures can be incredibly challenging, even for the most seasoned structural biologists, with all the possible configurations and folds.
Absolutely. It's like trying to predict how a piece of origami will fold based solely on the pattern of creases.
And with RNA, you've got all sorts of other forces at play, chemical modifications, interactions with ions in the surrounding environment that can influence how it folds.
Exactly. So while AF3 might not have cracked the RNA code entirely just yet, the fact that it can even tackle these structures with a reasonable degree of accuracy is impressive in itself.
It speaks to the power of these deep learning approaches to extract patterns and make predictions, even when dealing with incredibly complex data.
It does, and it's not just about overall accuracy either. The research has also delved into how well AF3's predictions align with its own internal sense of confidence, how sure it was about a particular prediction.
Because that's crucial, right? It's one thing to have a model that spits out predictions, but knowing how much to trust those predictions is a whole other ballgame.
Precisely, and that's where these confidence measures that are built into AF3 come into play. The research suggests that these confidence measures are actually well calibrated.
Meaning that if AF3 assigns a high confidence level to a particular prediction, users can be reasonably sure that the prediction is accurate.
Exactly. It's not just a guessing game. There's a solid statistical foundation underpinning those confidence scores.
Which is incredibly valuable because it allows researchers to focus their efforts on the predictions that are most likely to be true.
It's all about maximizing efficiency, both in terms of computational resources and in terms of guiding experimental validation.
So we've talked about benchmarks, we've talked about confidence measures. What else did the researchers do to really put AF3 through its paces?
They didn't stop there. They wanted to explore its performance on a wide range of biomolecules, including those with covalent modifications.
Ah, those chemical additions that can significantly impact the molecule's structure and function.
Exactly. Things like glycosylation, for example, where you have sugar molecules attached to specific amino acids on a protein.
Those glycosylation patterns can be really important for how proteins function and interact with other molecules, right?
Absolutely, and they can be a real pain to predict because they often involve complex interactions and can significantly alter a molecule's shape.
So how did AF3 handle those covalent modifications? Was it able to accurately predict their positions and how they influence the overall structure?
It did, surprisingly well, actually. They found that AF3 could accurately predict the positions of these modifications within the overall structure, which is a significant feat.
Most tools would probably stumble when faced with those modifications, treating them as outliers or ignoring them altogether.
And that's why AF3's ability to incorporate those modifications directly into its predictions is so important. It's providing a much more complete, a much more accurate picture of these biomolecular interactions as they occur in nature.
It's like the difference between looking at a blueprint of a building and actually
Yeah.
walking through the finished structure.
That's a great analogy. You might get the basic layout from the blueprint, but it's only when you see the details, the fixtures, the finishes, the way the spaces flow together, that you truly understand the design.
And those details, those covalent modifications in the case of biomolecules, they can make all the difference in how those molecules function and interact with their environment.
Precisely. They're not just decorative flourishes. They're often crucial for the molecule's activity and how it interacts with its surroundings.
So to sum it up, AF3 not only predicts structures with impressive accuracy, it does so while accounting for a wide range of biomolecules and modifications, and it even provides a measure of its own confidence in those predictions.
That's a pretty good summary. It's a powerful tool, and the research really highlights its potential to change how we approach everything from drug discovery to understanding the fundamental mechanisms of life itself.
It's clear that AF3 is a game changer, but let's delve into those potential applications a bit further. Where do you see AF3 making the biggest waves in the near future?
Well, one area where I think AF3 is poised to have a profound impact is drug discovery.
It's exciting to consider the potential impact AF3 could have on expediting drug discovery.
It really has the potential to revolutionize the field. Imagine being able to screen thousands, even millions of potential drug candidates in silico using AF3 to predict how those molecules would interact with a specific drug target.
That would be a game changer, especially for those diseases that currently lack effective treatments.
Absolutely, and it's not just about speed. By understanding precisely how a drug molecule interacts with its target, we can design more targeted therapies, drugs that are more specific in their action, minimizing those off-target effects that often lead to unwanted side effects.
It's about moving away from a trial and error approach to drug design, which can be incredibly time-consuming and expensive, and moving toward a more rational, targeted approach.
And AF3 could be the key to unlocking that more rational approach.
It's like moving from shooting arrows in the dark to using a high-precision laser to hit those disease targets with pinpoint accuracy.
I like that analogy. It really captures the essence of what AF3 could enable. And it's not just about designing new drugs from scratch either. AF3 could also be incredibly valuable for repurposing existing drugs for new indications.
That's right, using AF3 to identify drugs that might already be on the market, drugs that are safe and effective for one condition, and seeing if they might hold therapeutic potential for other diseases.
Exactly. We could potentially tap into this vast library of existing drugs and repurpose them for new battles, which could significantly accelerate the development of new treatments.
That would be a game changer, especially for rare diseases or neglected tropical diseases where the financial incentives for developing new drugs can be limited.
Absolutely. It's about working smarter, not harder, maximizing the resources we already have.
So AF3 has the potential to revolutionize drug discovery, but its applications extend far beyond developing new therapies, don't they?
They absolutely do. AF3 could also be transformative for understanding complex biological processes. Imagine being able to accurately predict the interactions between all the proteins and other molecules within a cell.
It would be like having a high-resolution map of the cellular landscape, allowing us to see how those intricate pathways and interactions govern cellular function.
And with that match, we could start to understand what goes wrong in diseases like cancer or Alzheimer's disease, where those intricate cellular networks go awry.
Those diseases are incredibly complex, involving a multitude of different genes and proteins.
Exactly. And to unravel that complexity, we need tools like AF3 that can provide a more complete picture of how those molecules are interacting and what's going wrong at a molecular level.
It's about moving away from treating the symptoms of a disease and moving toward a more precise, proactive approach that targets the underlying molecular causes.
And AF3 has the potential to accelerate that transition to a more personalized, mechanism-based approach to medicine.
But as we've alluded to throughout this discussion, while incredibly powerful, AF3 isn't a magic bullet. It has its limitations.
Right. Of course, as with any tool, it's essential to understand both its strength and its limitations.
We discussed earlier how AF3 might occasionally generate structures with slight stereochemical violations like incorrect chirality or overlapping atoms.
Right, and while those instances are relatively rare, they highlight the importance of not blindly trusting the model's output. Experimental validation is still crucial.
It's about using AF3 as a guide, a powerful tool for generating hypotheses and guiding experiments, but not as a replacement for those experiments.
Exactly. AF3 can point us in the right direction, but we still need those experimental results to confirm and refine its predictions.
Another limitation we touched upon is AF3's focus on static structures. It provides a snapshot of a molecule in time, but doesn't capture the inherent dynamism of biological systems.
And that dynamism, that ability of molecules to move and change shape, is essential for many biological processes. An enzyme, for example, might shift between different conformations as it catalyzes a reaction.
So while AF3 provides incredibly valuable structural information, it's not the whole story. We need to complement it with other approaches that can capture that dynamic aspect of molecular behavior.
And there are exciting advances being made in those areas as well, developing new experimental and computational techniques that can capture those molecular dances in action.
It's about integrating AF3 into a broader toolkit for understanding biological systems, using it in conjunction with other tools and approaches to gain a more holistic view.
Exactly. It's like any complex problem-solving endeavor. You need multiple perspectives, multiple tools to fully grasp the intricacies of the system.
And with AF3, we've added a very powerful, very precise tool to that toolkit, one that has the potential to accelerate our understanding of the molecular world.
Absolutely. And with that deeper understanding comes the potential for developing new treatments, new technologies, and a deeper appreciation for the complexity and beauty of life itself.
Well said. It's been fascinating to delve into the world of AlphaFold 3 with you. Thank you for joining me on this journey.
It's been my pleasure. It's not every day we get to discuss a technology with such profound implications for the future of science and medicine.
Indeed. And as we continue to explore the capabilities of AF3, it's crucial to do so with a sense of responsibility, ensuring that we harness its power for the benefit of humanity.
Well said. With great power comes great responsibility, and it's a responsibility we all share as we venture further into this exciting new frontier of biomolecular research.
To our listeners, thank you for joining us on this deep dive into AlphaFold 3. We hope you found it as fascinating as we did. Until next time.
Thanks for listening to Mythos AI. Stay tuned for more concise insights. And remember to hit the like button and subscribe for future episodes.